Then you can have a mix of several genes and environment, like the common disorders, asthma, diabetes, and then things that are mainly environmental, like infectious disease, although there are genetic components determining how susceptible you'll be and how you'll respond. And finally, chance events, things like trauma or environmental things, struck by lightning, lead toxicity, and so on. For all these serious childhood disorders, genetic diagnosis is available. We can get it done, and for most of the common disorders, that's available here in South Australia and with minimal fuss. But for the uncommon disorders, which are uncommon, but for each family that has them, are extremely significant, testing is not so easy to obtain, and we usually have to send samples overseas, which costs a significant amount of money. But I think the thing is to realise that when you study an individual with a single gene disorder and you find the gene abnormality, that information that you've gained is then usable in a number of ways which are very cost effective. You can use it to test other members of the family to see whether they've got the genetic disorder or are a carrier, and also you can use it, having done that, to perhaps undertake strategies to prevent the disorder and save the health system a considerable amount of money. And a good example of that would be familial cancer, where it takes a lot of money to find a familial breast cancer gene to begin with, but then it can be used to sort out who in the family has inherited it and not, and susceptible to breast cancer or not, and then very effective strategies can be implemented to address the problem. It's interesting that diagnostic technology is changing really quite rapidly, and we've moved in the last few years through a whole range of technologies which are very well illustrated in the next slide for the diagnosis of intellectual disability. Now, intellectual disability is common in childhood. It's 2 to 3% of all children, but sadly we don't identify a cause in at least half of children, even with quite significant intellectual disability. And there are many gene and chromosome abnormalities that cause intellectual disability, but unfortunately each accounts for only a very small proportion of all cases. And so you need a surveillance or screening strategy to try and deal with this. And if you look back to the 1960s when chromosome testing became available, that was the screening test. You studied a person's chromosomes and in a proportion, for example in Down syndrome and some other well-known syndromes, you would find an abnormality. But it's a very crude test where you see only abnormalities in number of chromosomes or large pieces missing or extra. Then along came a technique called fluorescence in situ hybridization, called FISH, where you could see that certain segments, if you knew where to look, were missing. As here, this chromosome is missing the signal that you see over here, and that was followed obviously by MLPA, another new technology that looks for variations in copy numbers at particular sites on chromosomes, and here showing a situation where a deletion is present where all the targets within the system for that region are at a lower level than the rest of the genome, suggesting that there's a deletion. We've had sequencing for many years, and most recently we have CGH array technology, which allows us to look in great detail at the whole of each chromosome for copy number variations, the situation where you might have a piece missing or duplicated. And these technologies have come along very rapidly, and we've had a great deal of difficulty. You introduce one technology and then almost immediately you're introducing the next technology, and it's quite a challenge, and there are obviously cost implications, and we have to talk to our masters in order to move from one to the other. But interestingly, as time has gone on, there may be a convergence of all these technologies, and it's been suggested that so-called new generation sequencing may be a way of doing what all these technologies do together, because not only in this type of sequencing do you get a gene sequence, but you also get a dosage or copy number, and therefore you do what this, and those things do. And ultimately it'll be very cost effective in terms of what we pay now. So technology is changing. We can attempt diagnosis with screening strategies, but very often we fail, and sometimes we simply fail because we haven't got the funds to send testing overseas in some families. Now, I'd like to move on to a different idea altogether, and that is the epigenetic control of gene expression. And what epigenetics refers to is the extra layers of instructions that affect gene activity without altering DNA sequence. And so you've got your DNA sequence, and there it is, but the system can modify that to determine how much of each gene is expressed. And there are basically two ways of approaching it that are common. One is to change the chemistry of DNA by attaching methyl groups at various points on the DNA, and very often that will silence a gene, or you can attach or change histones, which are the proteins around which DNA is wrapped, and affect the expression of the genes that are associated with those histone proteins. I talked earlier about imprinted genes, and imprinted genes, when we talked about them being silenced, are epigenetically silenced by these sorts of mechanisms. And it may be that regulatory genes work through epigenetic mechanisms as well. But of particular interest, I think, for the purposes of tonight, are the epigenetic effects of the environment on our genes. And so, perhaps to start, we can consider how genes are modified by the periconceptional environment. I think we've learnt about this in recent years, that the babies born from IVF treatment are at increased risk of a disorder called Beckwith-Wiedemann syndrome. And these are children with a large tongue, who are large overall. They may have an abnormality in their abdomen with the bowel protruding, but they're usually of normal intelligence, and they have a few other features. And our understanding is that this disorder occurs perhaps 4 times as often, but still rarely, in babies who are conceived by IVF. Now, it's not almost certainly the IVF treatment itself or the ICSI procedure that inseminates the ovum, but rather the composition of the medium in which the egg and sperm are treated that in some way adjusts the genetic information of the embryo at that very early stage so that the baby develops Beckwith-Wiedemann syndrome. The same may be true for Angelman syndrome, which I showed you a picture of earlier. Now, if we go on and talk a little bit about the intrauterine environment, I just wanted to talk about the Barker hypothesis. David Barker, in the mid-1980s and 1990s, developed this fetal origins hypothesis, and to be quite honest, I think we're lucky to have Geoffrey Robinson with us tonight, because Geoffrey was doing seminal work in sheep here in Adelaide that provided a lot of the basic data and scientific evidence for this human information that David Barker produced. And what David Barker observed was that low birth weight is associated with a number of disorders: high blood pressure, coronary artery disease, diabetes, hyperlipidemia. So, and what David assumed was that, look, while the fetus has a developmental agenda which is determined and driven by its genes, it actually can and must adapt to its environment, particularly in relation to oxygen and nutrient supply, if it's going to survive. And that adaptation influences size at birth, but has longer term implications. And the question is, and I don't think we understand fully why it happens, but how does the fetus adapt? At least one component seems to be a permanent change in gene expression that influences the chance of certain disorders in adult life. So what's the evidence? Well, there is some really good evidence from a number of sources. Firstly, the Dutch famine was shown to have long-term effects. The Dutch famine occurred during the Second World War in 1945-46, and it was a short, sharp famine where people went from reasonable nutrition to very poor nutrition back to reasonable nutrition